Our next session is on the topic of gene editing, where we will be joined by 3 researchers to give us an overview of gene editing, followed by an open Q&A with the community. So if you have questions, please submit them to the comment box below the live stream. So joining us today, we have Dr. Eric Olson from UT Southwestern, Dr. Charles Gershbach from Duke University, and Dr. Melissa Spencer from UCLA. So we're going to kick it off with Dr. Olson. So Eric, whenever you're ready, you can take it away.
I'm ready. If we could have the first slide put up. Good evening. I'm happy to be joining you from Dallas tonight to talk to you about recent work on gene editing as a therapy for Duchenne muscular dystrophy. These are my disclosures. Next slide, please. All of us share the desire to find a true cure for Duchenne muscular dystrophy, DMD, and this has posed many challenges, which are summarized here. The massive size of muscle, which comprises 40% of the human body mass, the massive size of the dystrophin gene, the largest gene in the human genome, the many different dystrophin mutations that cause DMD, the need for muscle and heart restoration to truly cure this disease, and the need for lifelong correction. Next slide, please. The dystrophin protein is schematized at the top of this slide, and the dystrophin gene is shown at the bottom with the 79 exons that splice together like pieces of a jigsaw puzzle to form the seamless RNA, which ultimately codes for the protein. Although there are thousands of mutations that have been identified in Duchenne boys worldwide, they cluster into hotspots. Next slide, please. Shown here in these 10 exons, which represent the majority of mutations, particularly deletion mutations in boys with DMD. This has led to the concept of exon skipping, in which various therapeutic strategies might be able to skip these out-of-frame exons and restore the open reading frame of the protein. Next slide, please. You can think about the dystrophin protein as a molecular shock absorber for all the membranes of muscles in the body, both skeletal muscle and the heart. Like a shock absorber, the central region of the protein is comprised of a series of redundant rod domains that function like coils of a spring, and the 2 ends of the protein, which are essential, anchor this protein in place beneath the membranes of skeletal muscle and heart. Next slide, please. There's a large number of patients with a relatively mild form of muscular dystrophy called Becker's muscular dystrophy, or BMD. These patients harbor in-frame deletions within the rod domain of the dystrophin protein and render it reduced in function, but it still retains residual function. And you can think about it as schematized here as a smaller shock absorber. It can work, but not as well as the wild type normal protein. And this has led to the concept of devising strategies to convert DMD mutations into BMD. Next slide, please. As all of you well know, there are numerous therapeutic strategies that have been developed and are continuing to be advanced to provide benefit to patients with DMD. In this session, we're going to focus on one of these. Next slide, please. And that is CRISPR gene editing, which is distinguished from these other types of therapies because it treats the underlying cause of the disorder, the genetic mutation in the DNA of the dystrophin gene, and attempts to correct or eliminate those mutations. Next slide. CRISPR gene editing is a revolutionary gene editing technology which was awarded the Nobel Prize this year. This is a 2-component system which involves a guide RNA shown on the left, which can be programmed to represent any sequence in the genome of any organism, including humans. Secondly, there's an enzyme often referred to as Cas9, which can then be guided to any sequence in the DNA by the guide RNA, and it will then cut the DNA. And when it cuts the DNA, it can eliminate mutations such as those responsible for DMD. Next slide, please. So you can think about the guide RNA essentially as a molecular GPS device. It can be programmed with high specificity to recognize sequences throughout the 3 billion letters of the DNA code in every cell in the human body. And it can deliver the Cas9 or the genetic scissors to that DNA target sequence where it can edit that sequence. Next slide, please. There are numerous efforts now to devise strategies to use CRISPR-Cas9 gene editing to remove mutations responsible for human disease. And these fall roughly into two categories. There are so-called ex vivo strategies in which cells can be taken out of a patient. This often occurs through blood cells, and these can be edited to remove mutations, such as those that might be responsible for sickle cell anemia, for example. And these can then be delivered back into the original patient and effectively cure the disease. Ex vivo gene editing cannot be adapted for Duchenne muscular dystrophy because of the massive size of the skeletal musculature and because of the inability to generate enough cells to colonize all the muscles and the heart. So this has required in vivo gene editing, in which the gene editing components must be injected directly into the body, and they must be able to find their target in the genome and edit it. And this is now being used for many types of therapeutic indications, including blindness, hypercholesterolemia, and as we'll talk about in this session, DMD. Next slide, please. Why does CRISPR lend itself to correction of DMD? Well, there are several reasons that are summarized here. The first is that the dystrophin gene, as we've said, is too large to replace by traditional gene therapy. Secondly, the dystrophin protein and gene have a modular structure that allows exon skipping, such that exons or segments of the gene that have mutations can be skipped, but can still restore the functional dystrophin protein. Third, the dystrophin gene resides on the X chromosome. Boys, which are predominantly the predominant DMD patients, have only one X chromosome, so this means only one gene needs to be corrected. Fourth, only a small fraction, a relatively small fraction of dystrophin protein needs to be restored in order to confer benefit to boys with Duchenne. And it's considered that even as little as 15% of dystrophin protein could have dramatic therapeutic benefit. And finally, skeletal muscle and heart cannot be reconstituted by stem cell transfer. And so this has necessitated strategies for in vivo gene editing. Next slide, please. This shows a representation of 5 exons of the dystrophin gene, exons 49 through 50, which splice together seamlessly to form the contiguous dystrophin protein, which is schematized at the bottom as as a shock absorber with coils of a spring. Next slide. The most predominant deletion mutation in boys with Duchenne is a deletion of exon 50. And you can see why that causes Duchenne, because when exon 50 is missing, the next exon in line, 51, does not fit together with 49, and this disrupts the protein. The next slide, please. So we've adapted a strategy in which we could deliver CRISPR to exon 51 with a single guide RNA. And by introducing a single cut in exon 51, this could induce either exon skipping to restore the reading frame by connecting 49 to 52, or to reframe the protein and provide for functional restoration of the protein. Next slide, please. We began by isolating blood cells from patients with Duchenne muscular dystrophy, and we could convert them into cardiac muscle cells in a dish, as shown at the bottom, and study their functions. And then we could correct them by delivering gene editing components to those cells, as shown on the top. In the left is cardiac muscle cells obtained from blood sample of a boy with Duchenne with no dystrophin. And on the right, we can correct it and restore dystrophin, as shown in red. This is an especially powerful approach because it allows us to test whether a particular guide RNA and a gene editing strategy can correct the mutation in that particular patient, and whether it can restore the function and expression of the protein in the actual patient. Next slide, please. So we've tried to adapt this technology to deliver it into animal models of Duchenne by introducing the gene editing components into a harmless virus, adeno-associated virus, AAV, which is familiar to all of you. Next slide, please. And we then asked whether the CRISPR gene editing components could not only find their way to the muscle cells that were affected, but could they deliver the components, and could they find the single mutation in the genome of all the muscle tissues of both mice and dogs. Next slide. So here you can see in the top would be dystrophin staining in representative muscle tissues of a mouse with Duchenne muscular dystrophy. In the middle, you can see a mouse. The top is the normal mouse. In the middle row is mice with Duchenne, and they have no dystrophin. And on the bottom, next slide, you can see the restoration of dystrophin protein after 4 weeks of one dose with the CRISPR gene editing components. Next slide, please. So what I've told you about so far is that you can correct Duchenne muscular dystrophy mutations by what we call single cut gene editing, in which one cut can correct multiple types of mutations by skipping exons. And this type of editing creates double-strand DNA breaks, and it can be imprecise in some cases. Most recently, there have been new strategies for gene editing that have been developed, such as base editing, which allows the conversion of a single letter in the DNA code to another letter to remove the mutation. And this is absolutely and highly precise at the target site. Next slide. Here you can see on the left is normal muscle from wild type mouse. In the middle would be muscle from a mouse with an exon 51 deletion. And on the right, you can see muscle that was base edited by changing a single nucleotide letter in the DNA code to restore dystrophin expression. Next slide, please. It's important to emphasize that strategies for correction by CRISPR-Cas9 editing result in the maximum size of the dystrophin protein that can be produced, as shown here, comparing at the top the normal protein with the gene-edited form of dystrophin that we've generated in the second to the top. In severe Becker muscular dystrophy, shown at the bottom, there's a truncated version of the protein, and the version shown at the bottom is correlated with very severe BMD. Microdystrophins represent an extremely truncated form of the protein that are being used in many promising therapeutic trials. And you can see that they're extremely small. They represent less than 1/3 of the level of the normal protein. Next slide. So what are the potential advantages, lastly, of gene editing as a strategy? One is that the corrected gene is expressed from the endogenous genetic locus. The second is, as I said, the maximum portion of the dystrophin protein is produced. And we believe the expression may be more persistent because it's coming from the endogenous gene and does not require continuous forced expression from an exogenous virus. Next slide, please. What are the potential challenges for gene editing? One is the potential for off-target genome editing. Might editing take place at a place we don't want it to occur? The second is their potential immune responses to Cas9. And lastly, the limitations to the efficiency of gene editing. Many of these are theoretical challenges, but thus far they haven't been shown to be major challenges to this technology. Last slide, please. So this slide illustrates where we started and where we are now. We've adapted this technology to correct mutations in samples from patients and have optimized it. And we've built mouse models to correct the mutation by AAV delivery and have then scaled up this work to correct it in large animal models. Next slide, please. So looking ahead, we're continuing to optimize editing and AAV delivery. We're continuing to demonstrate safety of the technology, and we're trying to adapt this to other monogenic disorders. And finally, my last slide just acknowledges the wonderful team we've had in Dallas that's been involved in this work with several of the key scientists and our collaborators at Vertex Pharmaceuticals. And with that, I'll hand it back to the studio and welcome questions later on. Thank you.
Thanks, Eric. We're just going to have a quick follow-up question. So you touched on a number of different strategies in that talk. Are there certain strategies that you think will be maybe more applicable for Duchenne, or do they all have their, or equally viable, just depends on the type of mutations that you're trying to address?
I think Duchenne, more than many other disorders, really lends itself to this type of CRISPR gene editing because it can be done simply by either changing a single base or one cut. And by skipping an exon or restoring it, you can bring back the entire massive protein, which is not possible by other microdystrophin strategies.
Great, thank you. Next, we're going to hear from Charlie Gershbach. So Charlie, whenever you're ready.
Thanks. Yeah, thanks so much for the opportunity to come and present, and thank you to Dr. Olson for a fantastic introduction for the broad field of genome editing for muscular dystrophy. As was mentioned earlier, I am at Duke University, but much of our work in gene editing for Duchenne has been partnered with Sarepta Therapeutics, who we've worked with and have generously supported the work that I'll be alluding to today. Next slide, please. So my role in this presentation was designated to talk a little bit more about what the challenges are in making gene editing a reality for Duchenne patients. But before getting into that topic, I just wanted to reinforce, I think, what we already heard from Dr. Olson, and I'm sure we'll hear from Dr. Spencer as well, that we all believe that gene editing has transformative potential for this disease, and we're incredibly excited about what gene editing can do as a treatment for Duchenne patients above and beyond other types of modalities that might be available. And that's exactly why we've all dedicated our careers to focus on this challenge. Next slide. And I just wanted to highlight, the diversity of approaches and models and laboratories around the world that are working diligently on this challenge of applying genome editing to Duchenne and having a great deal of success in these preclinical models. And so between the work of the three presenters tonight, as well as many other groups, only a subset of which are showed here, there's a great reason to believe that gene editing will someday have potential for treating this disease. Next slide, please. And along with that, I want to just build on what Dr. Olson already highlighted, which is that there are many different ways with which gene editing can be applied for Duchenne, whether it's targeted insertions, whether it's single cut for splice site disruption or reframing, or double cut approaches to remove one or more exons that can address a broad group of patient populations. There are a number of exciting approaches. And then as the field continues to grow with even next generation genome editing technologies like base editing and prime editing, as Dr. Olson highlighted, I think that we are continuing and will continue to see new exciting ways to manipulate the underlying DNA sequences to treat diseases like Duchenne. Next slide, please. And I think collectively, it's the diversity of approaches, the success in the preclinical models, and the efforts of groups like my colleagues on the call tonight that has generated, I think, this widespread enthusiasm for gene editing for Duchenne. Next slide. So with that in mind, I think that the question that I imagine many of the families in the audience are considering is, what are the challenges for making gene editing a reality, and what is the work, given the widespread success and attention in the preclinical models, what is it that all of us are still working on in order to bring this forward for Duchenne patients? And so really all of this boils down to two different categories, I think both safety and efficacy, and some of this was already touched on by Dr. Olson, and I'll go into it with a little bit more detail. Under safety, this includes the specificity and the hypothetical risk of off-target editing, precision of the on-target edit and controlling gene editing outcomes, and then tolerability of the treatment and the response of the human immune system to gene editing and gene editing machinery. And then under efficacy, of course, the challenges, as always with this disease, are around delivery, but also with maximizing efficiency of editing and durability of effect. Next slide, please. So first, to address the issue of specificity, as I think everyone understands, gene editing works by changing underlying DNA sequence. And so the hypothetical concern that is getting a lot of attention is, what if the gene editing machinery accidentally modifies the wrong sequence? And I want to really underscore this next point, which is this remains a hypothetical risk. Even though we know that it is possible under some circumstances in unoptimized systems for gene editing machinery to create off-target mutations, to my knowledge, there is no evidence to date of any adverse events in any preclinical model or in any clinical trial of something adverse happening as a result from an off-target gene editing event. So that being said, I think the question is probably, why are we wringing our hands over this possible off-target editing if it has not yet emerged as a risk in one of these preclinical or clinical trials? And that really boils down to the technical challenges of monitoring this problem and ensuring that it is not happening or could not potentially happen. And to kind of underscore the scope of the challenge, if we think that, in this type of treatment that is addressing all of the muscle cells in the body, which may make up trillions of cells, and in each one of those cells, there are billions of base pairs of DNA, the ability to even measure what the potential off-target DNA modifications might be is daunting. If you take all of those trillions of cells and multiply them by all of the billions of base pairs of DNA in each cell. And so one of the challenges that the field is working through is what are the right models to even understand what the risk is and what the possible frequency of off-target mutagenesis might be. And whether, even if there is off-target mutagenesis, whether that even has the potential to lead to adverse events, and how long we should wait. Because obviously, as many know, in the early days of gene therapy, in some cases, it was years before potential adverse effects from genomic modifications emerged in a patient population. Of course, at the end of the day, we're all, the field and collectively with the regulatory agencies is going to need to decide what is the acceptable level of risk, because it's simply not going to be feasible to be able to guarantee that all of the billions of base pairs in DNA across all of the trillions of cells are not changing. And in fact, because our genomes are actually changing all the time, every day, with every cell division or at any DNA repair event, that it's actually even not feasible to kind of comprehensively measure this effect. So, but still, work ongoing in the field to better understand this underlying issue. Next slide, please. So the topic of specificity on the previous slide refers to what the potential is to modify the genome at some other location. So in this case, it would be modifying the genome at somewhere other than the dystrophin target site. This slide refers to a distinct issue, which is even at the on-target site, how well can we control what the gene editing outcome is? And the reality is that almost all of these technologies, while largely create the intended edit in the dystrophin gene that we're looking for, many of them have the potential to generate other types of edits at the on-target site. And it's one of the things that the field is grappling with is understanding what the consequences are of what we call those unintended edits. Again, I want to underscore importantly that this remains a hypothetical risk. There is no evidence to date that any of these other types of modifications to the dystrophin gene could cause any type of adverse event. You might imagine that in this disease, the inherited mutation is a broken dystrophin gene, and changing a broken dystrophin gene to another variety of a broken dystrophin gene may not actually have any type of adverse effect, but it's something that I think that we want to be able to understand and be able to predict and ultimately be able to control. And so with that in mind, there's a great deal of effort in the field to work with these next generation editing tools like base editing, as Dr. Olson highlighted, to be able to better predict and understand what the editing events are that come out of this type of treatment. And again, I think we all believe that there will be some amount of acceptable level of risk given the challenge of being able to predict the editing event in all of the treated cells. Next slide, please. And then lastly, under safety is the issue of tolerability. So importantly, these CRISPR systems are derived from bacterial proteins and engineered RNA molecules. And so these are foreign to the human body. So just like an AAV capsid, of which I'm sure you've heard much about, these are recognized as foreign by our immune system. And we know from animal studies that at least in preclinical models, that the mammalian immune system can recognize these Cas9 proteins as foreign, just like they recognize AAV capsids as foreign. A additional complicating factor, just like pre-existing immunity to AAV capsids, that is the result of naturally occurring exposure out in the environment, the same is true for some of these CRISPR systems that exist in bacteria that we may be normally exposed to just in everyday life. And so there is evidence that some humans already have pre-existing immunity to the CRISPR systems, not unlike pre-existing immunity to AAV capsids. So this is an issue that, again, we're trying to better understand what the consequences may be, what the right models are, and what are the right assays, and how it might be minimized or controlled. Again, I don't think that there's evidence today that this is going to preclude CRISPR gene editing in humans, just like things like AAV gene therapies originally had challenges associated with immune response, but various efforts have been able to address those immune responses to move forward. I think similar things are going to occur in this field. I'll just highlight that there are ongoing clinical trials in humans in both the retina and the liver using CRISPR in vivo, and those will inform future studies in muscle and heart for diseases like Duchenne. Next slide, please. And then under efficacy, I'll just highlight, and I think Dr. Olson already mentioned each of these. There's always the question in a disease that affects as much tissue mass as all of the skeletal and cardiac muscle, the ability to get to all of the tissues. Largely the gene editing field is taking advantage of what's already been shown to us in conventional gene therapy studies and leveraging things like AAV vectors. And so as those fields continue to mature, they will inform what we do with gene editing. And we will continue in the gene editing field to take advantage of next generation viral and non-viral vehicles. Additionally, there's the issue of even once you get to the tissue, you now need to also hope that or rely on the fact that CRISPR is going to go and edit the target site. And so we need to make sure that we're maximizing editing to all of the cells that the CRISPR is delivered to. And there is the question of durability. And while gene editing is permanent to the DNA of a cell, because muscle is a tissue that does turn over, we need to ensure that those edited muscle cells also persist, which also asks the question of whether muscle stem cells are being edited, which I think is another important goal of the field. Next slide, please. And finally, as a conclusion, I just wanted to highlight that above and beyond the efforts of my colleagues on the call and others that I highlighted earlier to specifically develop gene editing tools for Duchenne, it is quite, as well as many of the companies that are supporting these efforts, it is quite of broad interest to transform these technologies to benefit for patients, including this somatic cell gene editing consortium that's sponsored by the National Institute of Health, which a few years ago put $180 million into dozens of different funded grants across many different institutions to address many of the exact challenges that I just highlighted in the previous slides. Next slide, please. And this just highlights some of the different institutions that are supported across developing new animal reporters and testing systems, new delivery vehicles, and new types of editing machinery. Last slide, please. And so with that, again, I just want to thank those that have supported our work, the students and postdocs that I have the privilege to work with in this area, as well as all of the patients and families that also support all of these efforts. Thank you.
Dr. Gershbach, that was a wonderful overview. You shared a couple of different challenges in efficacy. Of those that you shared, delivery, efficiency, durability, which do you see as the biggest challenge as we look towards future trials in Duchenne?
Yeah, well, I think that one of the most promising, where a lot of the promising outlook comes from gene editing is from some of the successes in the gene therapy trials. And so, I think that the gene therapy trials have shown to us that delivery is possible, that we can deliver these AAV vectors to the skeletal and cardiac muscle. And I think that we also know that that can be durable. And so I think that the key challenge to me under efficacy would be making sure that the editing efficiency is high enough, because we know that we can deliver the gene therapy. The AAV trials have shown that we can deliver to the tissues. And so I think that maximizing efficiency once we deliver that gene editing machinery is going to be the key aspect of once we scale up to larger animals beyond those that we've been treating.
Great, thank you. And finally, we have Dr. Melissa Spencer joining us as well. So Melissa, whenever you're ready.
Ready. Thank you. Thank you, Ryan. Thank you, Eric. So you've heard prior to my talk from Drs. Olson and Gersbach about the promise of gene editing for Duchenne muscular dystrophy. And my job now is to talk about how we deliver those gene editing components to all muscles of the body. So next slide, please. So delivery of gene editing components or for gene replacement therapy is one of the most difficult problems we face in the field of muscle gene therapy. But luckily, nature has provided us with a solution to this problem, and that is the adeno-associated virus, which Dr. Gersbach just mentioned. So this is a naturally occurring virus. It's non-pathogenic in humans. And in order to use it to deliver our gene editing components, also microdystrophin, all the viral genes are replaced by our cargo that we're trying to deliver. And what you're seeing here is an AAV molecule that is decorated with a lot of different proteins that are shown in different colors. Can I have the next slide, please? And these are the capsid proteins. So AAV is encapsulated by these capsid proteins, and it is also made up of a single-stranded DNA molecule. So there's two components to the AAV system. Next slide, please. And it's the capsids that serve like a zip code. So they help target the AAV that we're using to the tissue of choice. And so what I'm showing you here, represented by different colors, are different capsids or different serotypes. That's how they're referred to. So for skeletal muscle, for muscular dystrophy, we want to target both the heart and skeletal muscle. Next slide. And currently, many trials are using AAV9 or RH74 for Duchenne muscular dystrophy. And so there are many advantages and disadvantages to using AAV. The biggest advantage is that it can be delivered throughout the body, through the blood, and efficiently target skeletal muscle. And the fact that it is FDA approved already for some applications like spinal muscular atrophy lowers the inertia for getting approval for Duchenne. There are some disadvantages, though. It does have a packaging limit, and so for some gene editing components like Cas9, SPCas9, sometimes we have to use two different vectors, and that effectively increases our dose. Or you could say reduces our dose that we're allowed to deliver, since we have to double the number of AAV particles to get a single packaging, a single platform in. And it is also limited to single dosing because of the immune response that develops. And I'm going to talk about that in a minute. This immune response can result in some adverse events. And we've seen some of these in some of the microdystrophin gene therapy trials. Next slide, please. And so this immune response develops in response to our immune system seeing AAV, which looks very much like a naturally occurring virus. And when the immune response develops, it develops of two types. So there can be the innate immune system or adaptive immune system. And these two components of immunity have a lot of interplay between the two. Next slide, please. And so innate immunity is what I'm going to talk about first. This is the first line of defense that our body uses to see pathogen. And it's based on pattern recognition. Next slide, please. So there are two, next slide, please. Sorry, I animated these slides. So there are two main components of innate immunity that are being seen by the human immune system. And these two components are toll-like receptor signaling and complement activation. You may have heard about complement activation that's occurred in some of the microdystrophin trials. Luckily, we have a way to resolve this with Solaris, which is a complement inhibitor, and this seems to be doing a very good job of reducing any adverse events. Toll-like receptor signaling does not seem to affect the safety issues of the AAV delivery, but it may affect the efficacy. So we also have ways to cope with TLR signaling that includes CpG depletion and using a TLR inhibitory sequence. So scientists are working on developing ways to inhibit this pathway, and it may help increase the efficacy of the therapy. So this is innate immunity. Next slide, please. There's also adaptive immunity. And this is the arm of our immune system that has some memory. It remembers if it's seen a pathogen, and then the next time it sees it, it develops a very strong, quick, and robust response. The main issue, or the main arm of adaptive immunity is humoral immunity, which is the antibody response. That is the limiting factor for AAV. And so when our bodies see AAV, similar to with COVID, we develop antibodies. Those are these little structures that I'm showing you in this picture. And these antibodies will decorate AAV and tell the rest of the immune system to eliminate it. So this is a big issue if we want to consider redosing. And it's also an issue because many of us have been exposed to AAV throughout our lifetime. We've been exposed to the wild type naturally occurring AAV, and it's estimated that about 50 to 70% of us have this exposure. So it's going to be very important that we learn to deal with the pre-existing immunity that many of us have, and to deal with the pre-existing immunity that develops from the initial dose. Next slide, please. So why do we want to redose CRISPR if it's a permanent treatment? Well, the reason is that AAV is not 100% efficient. Not all fibers are transduced from the first dose, and not all nuclei are edited. And we also know that even if we had 100% transduction, 100% editing, we would still lose some of these edited nuclei over time with growth and with muscle repair by non-edited muscle stem cells. So when there's slight damage to the muscle stem cells come in and they repair it, and they could potentially replace some of these edited nuclei. Next slide, please. And so by redosing, we can retain the edited nuclei by continuously, maybe not continuously, but by being able to replace those lost edited nuclei periodically. Next slide, please. So there's a couple ways that we could achieve redosing. One is plasmapheresis, which just involves removal of the plasma and replacing it with new plasma that does not have those pre-existing antibodies. This is very possible. It's been done in primates, and it's been demonstrated that redosing can occur after using this type of approach. Next slide, please. Another is to use this enzyme that degrades the antibodies that are pre-existing. It's been demonstrated in this paper that this can be accomplished. It's an enzyme that's actually in bacteria. It can be applied prior to administration of the gene therapy. And this is currently being tested in preclinical studies, and I think it will probably go to clinical trials soon. Next slide, please. And the last is to block the generation of these antibodies prior to dosing. And so Barry Byrne and Manuela Corti have shown that a specific immunosuppression regimen, if given prior to the first dose of AAV, can actually allow for redosing. Now, they've done it only by intramuscular injection, but it seems to be working. We've done it in animal models. It works very well for systemic administration. And so this is the third way in which we could achieve redosing. And it's likely that we would need to use all three of these methods together, or two, to have a robust redosing protocol. Next slide, please. And so I've mentioned redosing, but the last manner in which we could improve the efficacy of gene editing is to target muscle stem cells. Dr. Gersbach mentioned this briefly. And so instead of redosing, if we had an AAV that could target myonuclei and muscle stem cells, we would have a very long-lasting, robust, and durable therapy, because these muscle stem cells can divide and repopulate their niche where they reside, as well as replace the lost nuclei that would have occurred through dilution of satellite cells. So I think that is my last slide. And I think we have to go back to the studio for our discussion. So thank you for listening.
Thank you, Dr. Spencer. Really appreciate that overview. I mean, I think it goes further to that conversation that we had about the lessons that we can learn from the current gene therapy trials and how that will be applicable to gene editing. Something that we have talked about a little bit is in the past, non-viral delivery. Is that, are you expecting that we're still going to see something like AAV used as the primary delivery tool for gene editing rather than something like non-viral delivery as we look towards the clinic?
Absolutely. I don't think we are anywhere near where we need to be for non-viral delivery. I haven't seen any evidence of that. My lab spent probably 3 years trying to get some non-viral nanoparticles to be able to traffic to muscle and traffic, across the sarcolemmal membrane. And no matter what we tried, we couldn't get it to work. So I think Dr. Olson has also had a paper relating to non-viral delivery. And so maybe he could comment, but I think right now AAV is the vehicle of choice. It's very efficient compared to any other delivery method.
Great, thank you. And we've had quite a few questions come in and not a lot of time. So of course, one of the big questions we've had come in from a number of folks is timing. And I don't think anyone necessarily on this panel can give definitive time. If you want to offer that up, I'm not going to stop you. But I think the question I'd like to ask you is, as we think about the current state of the science, is there any question you feel is really important for us to answer before we move into dosing a Duchenne population where we have to do this systemically? And Dr. Olson, I'll maybe start with you.
But you can also touch on timing too, if you'd like.
I think that the timing is always a big issue with a new therapy like this. I think one of the big challenges that the entire field is facing, not just the gene editing folks such as us, but others, is virus production and the types of adverse effects that some of these high doses of the viruses have yielded. So I think to the extent that virus manufacturing can be improved with respect to efficiency and doses can be lowered, I think that's going to really benefit things. I think it's also important to point out, as we did, that gene editing could be a one-time correction because muscle cells last for a very long time, and we hope that once the correction's made, that will be sustained.
Great, thank you. And Dr. Gershbach, I think you kind of already alluded to maybe the one you thought was the biggest, but if you have any others, I'd be happy to hear them.
Yeah, I think, you asked earlier about my thoughts about efficacy. I also point out that I think that just understanding how the immune system is going to respond to these things, I think is critical. And, I think that the major challenge there is that we know from gene therapy studies and others that what was seen in animal experiments and preclinical studies didn't necessarily predict everything that was seen in humans. And so I think that understanding what the right systems are for predicting and understanding how the human system is going to respond to these is kind of a critical issue for the field.
Great, thank you. Dr. Spencer?
I agree with both Dr. Olson and Dr. Gershbach regarding the being able to predict off-target immune responses, et cetera. We really need better systems, especially because the mouse has not been as predictive as we would have liked for the human immune system. So I think a lot of comparisons need to be made to know what we can model in the mouse that would predict. I think also learning or identifying biomarkers that we can screen for in patients prior to dosing that would predict, for example, whether they need to have a complement inhibitor or a different type of inhibitor. I don't think there is a really robust and reproducible way to do this screening right now. Every trial does different pretrial screening, does it slightly differently, has a slightly different cutoff. we're looking at a limited number of immune targets. So right now, it's looking at binding and neutralizing antibodies and looking at interferon gamma responses in T cells. And that's probably not sufficient. I think we need to better understand how the immune system sees AAV, what cells are getting activated. I think targeting some of these cells might improve not just safety, but efficacy for the long term. And I do agree with Dr. Olson that if we have a very efficacious therapy from the beginning, redosing may not be essential for 10 years. It is not as if it's going to be a weekly or monthly thing, but there will likely be a point where you would want to redose if you started with a baby and eventually that baby's going to grow into a full-grown adult. You're going to probably need to redose at least once. So I still believe that giving immunosuppressant at the start of the treatment is going to be critical.
So I know that the timelines are hard, but I am going to ask because I know what our families really want to know. Are we talking over 5 years or in under 5 years that we could see patients dosed with gene editing?
I sure hope it's in under 5 years.
Okay.
Yes.
I guess, and as a follow-up to that, because we've had questions about different mutations, deletions, point mutations. Dr. Olson, are there any, I guess, mutations where it's more challenging or maybe not feasible for gene editing, or do they all, have the potential to work with this strategy?
This is a really good point that I'm glad you brought up. There are types of mutations, particularly large deletion mutations in the dystrophin gene at either end, at the 5' end or the 3' end, that remove essential domains. And if that region of the genome is gone, there's nothing there for CRISPR to edit. So fortunately, those represent a relatively small fraction of the total number of Duchenne mutations. But yes, those would be more amenable to other approaches.
Great, thank you. And Dr. Gershbach, you had mentioned, in your introduction about insertions. And so we had a question come in about, is there, a limit to the size of an insertion that could potentially be done with gene editing? Are there, maybe ideal deletions that would be targeted?
Yeah, I think, to the point that Dr. Olson just made, hypothetically, gene editing can do a lot of different things, right? It can delete things, reframe things, disrupt things. It can also insert things. And we've worked in that area as well. But, I think the challenge is that they don't all work equally well, right? Some are harder than others. Some are more efficient than others. Some are less precise than others. And so, there's a reason why I think everyone on this call has focused on things like removing exons or disrupting splice sites to create these truncated dystrophins, as Dr. Olson highlighted, because that's kind of where the lowest hanging fruit is for the gene editing technology. Things can be inserted, but it's harder to insert. It's less efficient to insert things than it is to remove things. And then as the insertion gets bigger, it also becomes more challenging and less efficient. That being said, there are some tricks that you can do to take, for example, take different exons and squeeze them, multiple exons and squeeze them together into one kind of master exon that can serve to restore the protein. But all of these things are going to be more challenging than your conventional just remove an exon. I will say though that the technology in general across all the different gene editing platforms is moving remarkably fast, right? Base editing was one example, as Dr. Olson highlighted, of something that we didn't think would be possible, 5, 6, 7 years ago, and now it's reality and even moving into the clinic. So for other disorders. So I think that I remain optimistic that the field and the technology is going to continue to mature, but that's going to take time.
I would just second that and say that there has never been, I don't think, a more exciting time in science when one can understand the error in the genetic code and can actually correct it. And we're not quite there yet, but the pace of this field is breathtaking and the potential is breathtaking. And so I could not be more optimistic about where this field is headed.
Great, thank you. And I think on that note, we're going to call a close to this session. We had a lot more questions. We would love to get to them. I think we would welcome an opportunity to speak with you all in the future. This was incredibly insightful and educational, and I think it was really helpful for our community. So we really appreciate you taking the time to speak with us tonight.
Wishing you Godspeed. Thank you so much.